And these are two more of my heroes. This is Muhammad Ali, former world champion heavyweight boxer. There are probably three people in the audience, other than me, who remember when he was Cassius Clay. And this is Michael J. Fox, a very famous actor. And both of these very strong men are victims of Parkinson's disease. Both have been quite vocal about it, and each is a fighter in his own right. And Parkinson's disease is just one of the examples of the tragedy when the brain doesn't work. And we should all really be incredibly grateful for the fact that our brains do work, and of course sensitive to any changes in how they're working. So how does the brain work? Well, the main element in the brain is a cell type called a neuron, and this probably is not news to most of you. A neuron has a cell body called a soma, and that cell body gives off lots of dendrites. And the purpose of dendrites are to collect information from other neurons. And then all that information is collected, assembled in the soma, and it's turned into action potentials. And action potentials are electrical events that then are transmitted down the axon to terminals where they affect other neurons. And what's unique about the neuron is that this axon allows information to be transmitted a long ways. And so neurons have action potentials, trains of action potentials, and they can communicate the information that's in those action potentials to other neurons at faraway places in the brain. Now, inside of neurons are molecules. And if you go to Google, at Google Images, and ask for molecular pathways in neurons, you find millions of hits. And you can take they're all different. You can take any one. This is one of the simpler ones. It just shows that there are lots of molecules. There are receptors to which chemicals bind to cause ions to diffuse through the cell membrane. There are pathways inside of the cell that have effects on the future function of the brain cell, and there are millions of them. It is really remarkable, and we know a tremendous amount about them. Now, I would contend that this is very important information, but it's not the information we need to understand how the brain works. Synapses are one place where the molecular mechanisms are kind of important to us in terms of how the brain works. And the synapse is the connection between one cell's axon and another cell's dendrite. And at synapses, chemicals are released when an action potential comes down. They diffuse across this very small gap. They bind to receptors on the postsynaptic membrane, and they cause a response in this neuron. And each postsynaptic neuron receives inputs from many presynaptic neurons, and that's how information is transformed inside the brain. Now, this is all for background. One more point, and that is that the brain is capable of learning, and we now know from work done in neuroscience that one of the main mechanisms of learning is changes in the way synapses work. And so in an initial state, this synapse has relatively few packages of transmitters and relatively few receptors, but after learning has occurred, it has more vesicles that contain more chemicals for transmitters and more receptors on the other side. And so when one action potential comes down, it causes a bigger response in the postsynaptic neuron. And we now know that that's the mechanism inside the brain that leads to learning in our behavior. Now, I think that the most important element of how the brain works is circuits. And one of my former colleagues, Howard Fields, once said to me, the basic building block of behavior is neural circuits. And so we need to understand how circuits work, how groups of neurons work together. Now, let's go back, come on. This is the hippocampus about which Liz spoke. And you can see from the Golgi stain, this is probably from Cajal, that there are lots of different kinds of cells in there, lots of different kinds of neurons, and that some neurons give off axons that go and contact other neurons, and there are and then again these guys contact other neurons. There are very specific architectures to these connections, And to understand how the brain does something, we need to understand those architectures, and we need to understand the signals that are sent down those axons and how they're integrated in neurons. So the brain works on many, many levels. In fact, it is integration across levels, from molecules to synapses to cells to circuits to behavior, that gives us the cognitive neuroscience that we embody in ourselves. So as I said, Howard Fields told me that circuits are the basic building blocks of behavior. And so my question is, how do circuits of neurons convert sensation into action? Okay, just one more tangent here. Why should we care about how the brain works? Well, there are two classes of answers to this question. And when I started in this field and when Dale started in this field, the answer was because part of our destiny is to understand ourselves. The world has changed a little bit, and I still have a passion for the part of neuroscience that helps us understand ourselves. But we now are told by the people who are paying for our research that we should care about how the brain works because knowing how it works in health is essential for understanding why it fails to work in disease. And my sort of way of thinking about this is that disease comes from some of those molecules inside the brain not working right. And sometimes those molecular malfunctions start very early in life, but symptoms don't appear for 10 or 20 or 40 or 80 years. So what happens? Well, molecular mechanisms are the fundamental problem, but they lead to circuit malfunction. And when we get circuit malfunction, then we get symptoms, just the same way that normal circuits generate normal behavior. So neural circuits are the link from the molecular mechanisms that cause disease to the symptoms that take us to the doctor. And so if we can understand that link, it's one very important piece in understanding disease, diagnosing disease, and ultimately in treating disease. In fact, one of my beliefs is that the brain's own learning abilities can be harnessed to help the brain heal itself. Now that's probably not in my generation's time in neuroscience, but it might be in your generation's time in neuroscience, and it's a very lofty goal. I should say it's a lofty goal that will be accomplished only if people like me and people like Liz and real doctors really work together. Okay, how do we discover how the brain works? Well, I think we have to start by defining simple and accessible behaviors to study. And just as Liz chose the simple and accessible concept of Euclidean geometry out of a whole panoply of possible, more complex concepts, I have studied eye movement, a simple behavior. And I strongly believe in the mantra of divide and conquer. Take something simple and break it down into its essential components. Break a simple behavior down into multiple components. Break it down into the different operations of different brain sites that are involved, and look at different times for different mechanisms and participation of different sites. I'm going to show you how we've used movements of the eyes with this kind of a divide and conquer strategy to understand something about how the brain works. And I'm going to assume that if we understand how we generate eye movements, we will have general principles that are used for all different kinds of behavior, all the way up to cognition. Okay, divide and conquer. We'll start by looking at saccades, and this is a video taken off YouTube, but provided by Neurokinetics. Neurokinetics makes eye movement monitors. And the first thing we're going to do is divide eye movements into different, do you want to do this for me? That would be great. Into different kinds of eye movements. And this is an example of saccades, and you can see that the subject is just rapidly changing the eyes from one place to another with fixations in between. The subject is being asked to look at a spot that you don't see, but is doing it with these very rapid changes. And if those of you who are really aficionados can see eye position as a function of time, these are the saccades, these are the fixations. Okay, the next slide shows smooth pursuit eye movements. And if you want to start this movie for me, this is me, and my task is to fixate and track this spot of light as it moves across the screen. You also can click right on the movie, and that will start it. Yep. And you can see that to do so, I'm generating smooth eye movements that look rather different from the saccades that I showed you on the previous slide. And these smooth eye movements require the smooth motion of this visual stimulus, and I'll show you that in the next clip, where I was instructed to try to move my eyes as slowly and smoothly as I can, but without the moving target, and you can see I'm generating saccades. So we have two different systems that respond, that look different in terms of the movement, and that respond differently to stimuli. Now, we are interested in understanding not only what the brain can do, but how it does it. And to understand how it does it, we need access to the circuits that generate the behaviors. And for that reason, we do our research in a preparation that's ideal both for the behavior we're studying. In fact, the monkey is the best eye movement species that exists, but also ideal for the approach that we need to use, which is to monitor the electrical activity of individual neurons. And so this is a picture of our preparation. This is a monkey. He's sitting in a plastic box that we call a chair. He has a head holder implanted on his head that allows us to secure his head to the ceiling of the chair in a way that is not distressing to either him or us. That means we can cause him to make eye movements instead of head movements. And we use a magnetic method to record his eye position. We implant a very lightweight coil of wire on the eye, and that coil is then carried with the eye as it turns back and forth, and we place the animal inside a magnetic field that's created by this cube. And those of you who, I think everybody probably has had high school physics, and so you may remember that if you put a coil inside a magnetic field, a current is generated in the coil. As the coil moves to different positions in the field, the amount of current differs, and we simply then have external electronics that sense the current and tell us what the animal's eye position is. The animals are trained. They're trained to fixate and track spots of light that appear on a screen in front of them, and in exchange for doing it correctly, they get fluids. Okay, you get to be the monkey. You'll get to be the monkey again, but relish this. The monkey's job is to fixate this white spot wherever it goes, and this is going to be a movie. And so when the movie starts, you'll see that the target will displace to the left and then ramp to the right. And you see what the monkey did. Can you run the movie again for me, please? You can see it again. Okay, here we go. Eye movement starts just before the target gets there, tracks beautifully, and stops when the target stops. Now, our monkeys will do this 2,000 times, which allows us in a day, which allows us to do quantitative studies of behavior. It enables what we do. What does this look like when you put it on a computer monitor or an oscilloscope screen? Well, these are records, and they show up here position as a function of time. And now you can see that the target was stationary and the animal fixated it perfectly. The target displaced and ramped, and in this example, the eye started to move in the direction of target motion. This is going to tell us that the animal is responding to the target motion. If the animal were responding to the position, he would have moved his eye that direction. So the smooth part of the response responds to the target motion, and then here's a saccade coming back, and saccade corrects the difference between target and eye position and brings the eye so it's pointing right at the target. Now, why is the animal motivated to point its eye right at the target? Because the part of the retina that has the greatest acuity, because it has the most receptors and the site where most of our color vision is in the fovea. And so we can better process visual images if we're pointing the fovea at the object that we want to see. Now, to study these, we simply take the first derivative. So we take position records and we differentiate them, and now you can see that what we did is we had a target that was initially stationary and then started moving to the right. In this case, it